You're ready. Go ahead.
Okay, so we're very lucky today to have Professor Julia Stephen, who's a professor of translational neuroscience at the Mind Institute, which is on the UNM campus, but for some reason is not integrated into UNM. I mean, is more closely associated with Lovelace. And the University of Colorado at Boulder. Anyway, she's going to talk to us about mammalian brains. And she's worked in this area since she got her PhD in physics. So she's a physicist doing physics rather than a doctor practicing medicine. She's published, she's been the author of over 120 published peer-reviewed articles in the subject of translational neuroscience and the use of magnetoencephalography to study the brain. That's a non-invasive way of studying the brain. Well, enough from me. Julia, please give us your lecture.
All right, thank you very much for the invitation. I will review a little bit about the brain and then talk briefly about some of the work that we have done to try and understand brain function and how that is related to behavior. So the brain is very complex, and this is just a picture of the brain that was collected some number of years ago now, but I like this picture just because it shows you kind of how much of a mess the brain is and how much of a challenge it is to really be able to understand what is going on in the brain. So this is just a small chunk of cortex where the astrocytes are shown in green, the neurons, which are the primary processing cells, are shown in red, and then the cell bodies are shown in blue. So how can we get information from this sort of messy picture? So we know that the human brain is very complex. It's estimated to have over 1 billion neurons, and each of these can connect to over 1,000 other neurons, which means that we have 60 trillion connections in the brain. This is a highly complex system that we're still trying to understand how to understand. The neocortex, which is 2 to 5 millimeters thick, is just the very outer rim of the brain, is sort of the computer part of the brain. And the white matter, which is the inner part of the brain, really consists of axons, which just connects those different regions of the brain and is really considered the wiring of the brain. So this is just one of the neurons that demonstrates that we have dendrites, and these are the sections that connect to other neurons, and then the information is passed along the axon to another part of the brain. It might be very close by, or it might be farther away in the brain. So the development of the brain obviously is a very complex process as well, because you go from having 0 neurons to 1 billion neurons in about 9 months. So if you just break that down with simple math, then that's averaging 3.7 million neurons a day that need to grow. And then that growth actually isn't consistent throughout those 9 months. And so at some periods, the rate is even faster than that. So this is just a really amazing process that the brain goes through. The very beginnings of the brain is what's called the neural tube, and that's what's shown here. It's the very first brain structure, and it's formed in embryonic day 20 to 27. So the process of how the brain is developed is also very complex. So within those 9 months, you go from, as I said, 0 neurons to over 1 billion. And the process is very well programmed. And what it does is it develops, just as a part of that initial part, it develops this ventricular zone, this VZ, And this is where the neural progenitor cells are located. They're the ones that actually generate new neurons. And the neurons migrate from that ventricular zone up. And it first lays down this purple series of neurons. And then as more neurons are generated, then it builds on different layers. And so then the darker blue and then the lighter blue, and they continue to move past their neighbors to develop what is shown on the right side of this slide. And the final version of our cortex, that neocortex that's 2 to 5 millimeters thick that I already spoke about, has 5 well-defined layers, and the white matter, which includes the axons, the wiring part of the brain, is at the very base of that, and closer to the surface is up here at layer 5. So after migration, axons extend from that initial location to other parts of the brain. And as I said, this can be very close by, or it can be quite distant, so centimeters. So these axons can be either short or long. And it's completed by gestational week 26. So in addition to adding lots of neurons, the brain actually goes also through cell death and pruning across development. And so most of the cell death actually occurs prenatally. So these layers that I talk about, the ventricular zone, and then there's another layer at the very top called MZ, that those are really layers that are there just to lay down the infrastructure of the brain. And once that infrastructure is built, then those regions really go away. And so cell death occurs in some cases. And what we're showing on the bottom are just the volume or the number of neurons in different parts of the brain. So frontal cortex, thalamus, and then over here is cerebral white matter, which again is relating to the axons or the wiring in the brain. So you can see in general, we lose the number of neurons over time. And this is across age in years. So you can see that we also lose neurons as we age. But what we're really focusing on right now is this very early part of development. So the other thing that you may have heard is that there's also synaptic pruning that goes on, and the synaptic pruning is really referring to how many connections those neurons make to other neurons. The synaptic pruning doesn't really, it's not related to cell death, it's just related to which neurons those neurons are connected to. And most of this happens really in the teenage years in terms of the synaptic pruning. So just as a little thought experiment, you can kind of think about the creativity of children and, the number of different things that they can kind of come up with in terms of, just in creative play and things like that. They have a lot more connections between different neurons than we do as adults. And this may really feed into the creativity that you see in children. So one way to also say this is that their brains are over-connected and are able to make novel associations. So if I just point out, so you can see that if you look at just gray matter, which is what these first 3 plots are showing, then you see this general decline. But if you look at white matter, which is the axons, and it's actually a much different pattern across age, across the lifespan. And this is zooming in on that a little bit. So this is now just showing the first 30 years of life. And we can see that white matter continues to develop through into adulthood. So why is the volume, in this case, it's actually looking at fractional anisotropy, but why do the properties of white matter change across development? And what's really changing in that case is these cells are being myelinated. And what myelination is it's a growth of this insulating sheath that encircles the neuron, and that sheath actually speeds transfer of information. So it's very similar to a cable. If you have an insulated cable, then you have faster transmission than you do with an uninsulated cable. So this insulation of neurons, of axons, continues throughout development into the adult years, and that actually changes the timing properties of the brain consistently through childhood. So this is just another pretty picture, really, but it really demonstrates that the development of the brain happens differently in different regions of the brain. But you can see that frontal cortex from 5 years of age is really what's changing rapidly. And this is really demonstrating that the blue is demonstrating that it's very similar to adult-like brain, and the red is indicating the areas that are not adult-like. So the areas that are changing rapidly are really frontal lobe. And if you look at some of the sensory regions, which I'll show in the next slide, but more posterior regions, those actually develop earlier. So these very basic processing develops at an earlier age than these higher cognitive processes. So what we're trying to understand is information processing in the brain. So we have this very complex system with lots and lots of neurons, and what we don't know is, how all of that works together in order to generate the complex behaviors that we can carry out as humans and mammals, generally speaking. So what we know very well is these very basic sensory processing. So if you think about your eye, we know that the eye is connected to the visual cortex back here in the back of the brain, and that's fairly hardwired. And then the visual cortex does a bunch of processing as well, and then that information gets transferred to the frontal lobe, and that's where you can depending on what you're trying to do, what do you want to do with your visual information that you just received? Perhaps you're trying to read, or perhaps you're trying to learn some new skill. All of the rest of that cognitive processing happens as a part of this brain network. Auditory processing is so the ear is connected to the auditory cortex, which is here on both left and right side of the brain. We know our tactile system is wired into this part of the brain. But the question is how do we take that input, which is obviously a key part of brain functioning, and develop these higher cognitive processes. So again, I've already mentioned that the neurons are the primary processing units in the brain. And so it's important to kind of understand how that works. So we have the cell body, the dendrites are what accept input from other neurons. The cell body takes all of these inputs from the different dendrites, and if it receives enough information, then it decides to fire off and sends off an action potential along the axon, and that then stimulates another neuron, and so on and so forth. And so you get this transfer of information from one neuron to another, from the cell body along the axon to the next cell. The synapse is connected, so this information actually occurs chemically. So when you have an action potential, and then at the very end of this process, that electrical stimulus actually causes the chemical signals to be released And those chemicals are then received by receptors on the other side of the synapse. And those chemical processes are then again translated into electrical signals on the other side of the synapse. So this is where different neurotransmitters and other chemical components in the brain are associated with brain function. And it's primarily at the synapse level. So I already mentioned about the electrical activity of these nerve impulses. So this is just a model of an action potential and what happens. And so if you get this input from different dendrites and you have a certain amount of really change in the electric potential within the cell, then you can cause what's called an action potential. And once an action potential is initiated, then it travels along the cell in a very stereotypical fashion. This is just another model of all the currents that are associated with this process of moving information along the neuron. So this is just another plot of an action potential. These are the different things that happen as a part of that action potential. So there are sodium channels that are opened and then sodium channels close, and then potassium channels open. And all of these are related to why you get an influx of ions into the cell, and then a decrease in those ions after a short period of time. So this action potential can move along the axon very efficiently. And the myelination that I've already spoken about actually helps to speed that along as well. So this is interesting, but if you actually look at the properties of this action potential, then the current that is generated is actually pretty symmetric in either going to the left or to the right. So if you're trying to measure electrical activity from a cell, the action potential is probably not actually where you're measuring it from, because the signals from this are mostly going to cancel out because of the properties of the action potential. What we also know in terms of electrical properties is that we have these things called excitatory and inhibitory postsynaptic potentials. And this is the electrical activity that happens on the other side of the synapse after the information has traveled along the axon with the action potential. And in particular, these excitatory postsynaptic potentials are asymmetric. So you see a very quick increase in the potential, and then a slow decrease. And because of this, you have this asymmetry in the currents that are developed in the neuron. And because of this asymmetry, then you actually have a current that is dominant in one direction. So if you put all this together, what we see is currents that are flowing along the axon. And then that information, those synapses connect to another cell body, either along the dendrites or directly on the cell body. And the electrical properties of that this is what you see for the axon, the action potentials, where you have the symmetric currents on both sides. And then postsynaptically, you see this asymmetric current. So because of this asymmetric current and the excitatory postsynaptic potentials, then you can actually get an overall magnetic field that results from that. So that's all fine and good, but you actually can't measure the magnetic field from a single neuron non-invasively, because it's still very small, right? I mean, we can't modify any anything with the strength of a magnetic field from a single neuron. So what you actually need is you need symmetry in the brain as well. And what is convenient, and this was discovered a long time ago. So this is one of the famous drawings from Ramon y Cajal from 1888, really showing us this is now neocortex. And what is being shown here are what are called pyramidal cells. And they're called pyramidal cells because they look like pyramids and they have what's called this apical dendrite. So all of these pyramidal cells have this single dendrite that goes towards the surface of cortex. And because of that, we get this current that is these currents that are all pointing in the same direction toward deeper into cortex. And because of that, you can get a stronger magnetic field if these are all stimulated at the same time. So what we think we can measure non-invasively with using magnetic fields is actually the graded potentials, these excitatory postsynaptic potentials originating from the apical dendrites of the pyramidal cells. You still need around 100,000 neurons to be simultaneously active in order to measure a magnetic field that is strong enough outside of the head to measure it non-invasively. So I showed you that picture at the very beginning that was very complex in the head, what are called astrocytes and neurons and cell bodies. So this is a picture of astrocytes. So the reason that we don't think that astrocytes are generating any signal outside of the head is because this is how they're organized. And if you look at the currents that are coming into the cell body, they're all organized in a random fashion. So within a cell, the currents will mostly cancel out, and between cells, the currents are also random. And because of that, we don't think that the signal that we can measure outside of the head is coming from astrocytes. And this is important because astrocytes are really there as support cells. They are not the primary neurons. They're not the primary cells that are actually transferring information from sensory cortex to other areas, but they are really, feeding the neurons and taking care of all the rest of the things that need to be taken care of in the brain in order to keep the brain healthy. So this is another visual just to kind of put everything together. So we have these apical dendrites from pyramidal cells, and in the human brain, our brain is folded. So you still have some variation in terms of the orientation of those currents, but you can imagine that this goes into here and it generates a current that is primarily pointing down into the brain. And you have, depending on what part of the brain is activated, you have what can be a larger current because all of these cells locally are activated. And from that, you can get a magnetic field, and you also get an electric potential from that. So one thing that I'd like to emphasize is that I've really been using the human brain in the figures that I'm showing you, but really a lot of our knowledge of the brain and how it works and the electrical properties and things like that all come from both animal and human research. So the question is like, why can we really translate information from animals where a rat has a much smaller brain than a human, and there are a number of studies that really emphasize the fact that the anatomical properties of gray matter are conserved across species. So we have approximately the same number of neurons within a certain volume, regardless of what species you're talking about. And this is critical because this means then that the electrical properties that we measure in the human brain translate to across species to all the way down to mouse cortex. So this is just one way of demonstrating that. So this is actually looking at a current dipole moment. So how strong is the current from activation within different cortices across species. So swine cortex. So if you look at the pig or the monkey, humans, patient 1 and patient 2, turtle cortex or guinea pig cortex, they're all within the same range in terms of how strong that current is from different stimulation. So what that means is that the neurodynamics that you measure in rat cortex versus human cortex should be similar. So I'd like to move into a little bit just talking about how we're measuring activity in the brain non-invasively. And I already showed a picture similar to this, but what we do typically is we provide some stimulus, and then we look at we measure the magnetic fields that are generated in the brain because of that stimulus. And we do that with this technique called magnetoencephalography. I already showed you a similar picture to this, but what we're doing is measuring that magnetic field generated by the currents in the apical dendrites. So the machine that we have looks like this, and what we actually have are these sensors that are inside this hard plastic shell. So this machine, in order to measure the magnetic fields that that are generated by the brain, we actually need to use superconducting quantum interference devices, or SQUIDs. So this machine is so big because it has a big dewar to hold our liquid helium in order to keep our sensors superconducting for measurements. And so these sensors are superconducting, and it allows us to measure very small magnetic fields generated by the currents in the brain. So MEG is measuring the magnetic fields due to the currents, but with a current, you also get an electric potential, which is what EEG measures. So EEG is actually much simpler. You can just attach some electrodes to the scalp and measure signals. It's not nearly as technically challenging from a sensor development perspective, but we have this other technology called MEG. So the question is why do we use MEG as opposed to EEG? And our primary motivation is because the electrical properties of the skull actually distort the EEG signal to some degree. So that's what these yellow lines are trying to demonstrate, is that because the skull has different electrical properties than the brain or the scalp, then it actually makes it difficult for the EEG signal or the electric potential to get through that barrier. Whereas with magnetic fields, they're only distorted by ferromagnetic materials, which typically our brains and our skulls are not made of. So because of that, the green lines, which are trying to demonstrate the magnetic fields, are not distorted in the same way. And so the advantage of MEG is that it's actually much easier to kind of figure out where the activity is coming from. So even though we measure non-invasively out at the sensor level, what we like to do is we like to use mathematical models to then reinterpret where that activity is coming from. But if you want to use those mathematical models, then you have to be able to account either for the physics of how you're distorting the signal in the case of EEG, or you use a technique that just isn't distorted, and then it makes your problem easier. So this is just a simple spatial demonstration. So if you look at the magnetic field patterns at the sensor level with MEG, this is stimulating auditory cortex with MEG and EEG, the same sources. With the EEG signal, you just can't, based on that spatial pattern, you can't even guess where the sources are coming from. And with MEG, it's between the two, the maximum and the minimum. So it's much easier to spatially identify where the activity is coming from. And then we have another complication during development between MEG and EEG, and that is that when infants are born, they have actually holes in their skull, right? So they have what are called fontanelles. And because of this, then it actually complicates the EEG signal even more because, with everything, things like to take the path of least resistance. And so this actually distorts the signal even more because the currents will try to sneak out through these holes rather than trying to force their way through the skull. And because of that, it makes it even more difficult to figure out where the activity is coming from with EEG. So what I'm going to focus on primarily today is talking about neural oscillations. In the physics domain, we're quite familiar with oscillations generally, but in real life, they're not perfect oscillations, but you can still, in a lot of signals, see remnants of oscillations. And that's what we do with our brain signals is, we just do a standard Fourier transform, and you can look at different frequency bands. So these are different frequency bands that have been identified that are related to different behavior. And a lot of the foundational research that that developed these different frequency bands was really based on EEG signals, right? Because you can still look at the same neurodynamics with both MEG and EEG. But we have these different frequency bands, delta, theta, alpha, beta, gamma, and those are from low frequency to high frequency. You can deconstruct the signal into these different frequency bands. And what we know is that different frequency bands are related to different behaviors. So alpha was one of the first signals that was measured with both EEG and MEG. And alpha oscillations that happened at about 10 hertz. If you close your eyes and you're not asleep, then your brain, your parietal occipital cortex will start generating alpha oscillations. And the interpretation of this now is that alpha oscillations are essentially trying to inhibit parts of the brain that are not supposed to be attended to, right? So the parietal occipital cortex is the part of the brain that processes visual information. If your eyes are closed, then it means that you should ignore visual information because there is nothing coming in. So it's a way for the brain to essentially say, ignore this, pay attention to something else. So many people are also familiar with time frequency analysis at this point. So oftentimes we use either wavelets or multi-tapers to look at little blips of oscillations because as you saw in the previous slide, we do see patterns of oscillations, but they don't happen forever. This assumption of Fourier transforms where the oscillation goes on forever is not true in the brain. So wavelets and multi-tapers have also been very useful for characterizing these neural oscillations in the brain. So this is just an example time frequency map from one of our studies. So what you can look at is this is time on the x-axis in milliseconds and frequency on the y-axis. And you can look at different frequency bands. So in this case, this was a multi-sensory stimulus. So we were presenting auditory and visual information. And one of the ways that has been hypothesized that the brain can put things back together, one thing that is quite interesting still is that the brain really likes to pull things apart and then put things back together. So I already said, the visual cortex, the information from the eye goes to the posterior part of the brain, and the auditory cortex, the information from the ear goes to the temporal cortex. Those are completely different brain regions, and yet we still see the world as very multisensory. So it's a really non-trivial problem to put that information back together. So the question is how does that happen? And one of the hypotheses is that neural oscillations are one way to sort of group things back together. And so we were looking at gamma oscillations in particular, which is this 30 to 50 hertz range, and looking to see if there were differences between our healthy controls and our patients with schizophrenia. And I'm not going to go into any more details about this study today, but it is important to appreciate that these different frequency bands seem to have different functions in the brain. And I just wanted to introduce the general idea of what a time frequency map looks like. I should mention, time zero here is when we presented the stimulus. So we're really looking at the activity in the brain after that stimulus is presented. So from time zero out to 500 milliseconds or half a second. So I had mentioned previously that we have a lot of consistencies across mammalian brains in terms of neurodynamics. And this was a study that looked at that specifically. So in theory, based on the electrical properties and the fact that we have the same number of neurons and the same amount of volume of cortex across species means that we should have the same electrical properties. But of course, it's always nice to see experimental data that demonstrates that. So this was a study that looked at humans using MEG and macaque monkeys, and this was an invasive study for the monkey study. So you can put electrodes in the brain and measure what are called local field potentials. Again, still looking at the electrical signal in the brain, but essentially using EEG rather than MEG, looking at the electrical potential. And then they also counted the number of spikes, so the single unit spiking from different neurons. And what you want to look at here is if you look at, this is just the raw signal that comes in, and then you can do frequency analysis on that. And with this particular stimulus is visual grading stimulus. In both humans and primates, we know that that generates gamma oscillations, these oscillations in the 30 to 50 hertz range. And you can see that there's this peak with the stimulus in gamma in both humans and the monkey cortex. And then you also see this increase in the spike firing. So because of this study, and this is not the only study, but this is just a nice example of it, we know that these gamma oscillations are related to spike firing and that they're similar across different species. And this is just another piece of that study. So they actually manipulated the visual contrast. So how different are the black and white bands in that visual grading stimulus. And if they're more different, then you get more gamma oscillations. And if they're less different, then you get fewer gamma oscillations. This is in humans, and you see the same pattern in the macaque brain. So again, it scales with the contrast of the stimulus. So this is just another study that really emphasizes the relationship between spike firing and neural oscillations, specifically gamma oscillations. So this black line is representing the spike firing, and the time frequency plot underneath that, you can see that those gamma oscillations are directly related to the spike firing in this study, this invasive study. So I want to reorient again back to human studies. So one thing, I've already given the example of alpha oscillations, and we know that alpha oscillations increase when you close your eyes, and this is just a demonstration of that. So if you look at the black line versus the red line in parietal occipital cortex, which is where this sensor is coming from, you can see that there's this big increase in the 10 hertz oscillations. And then you also see manipulation in alpha oscillations depending on whether you move your hand or not. So there's a similar oscillation that happens in sensory motor cortex. If you are resting your hands, then you get this oscillation. And when you activate your hands, then it makes that oscillation go away. So this is just more experimental evidence that suggests that these neural oscillations are related to behavior in the brain. We also know that there's some coherence between our MEG signal, and EMG is measuring the muscle electrical activity. So if you look at the muscle electrical activity in the hand, then that is also correlated with brain activity. So again, this is another demonstration that that behavior, moving your hand or not moving your hand, is related to the neural dynamics within the brain. So another thing to look at, we also know that there's this phase amplitude coupling, and what phase amplitude coupling means is if you look at low frequency oscillations, then we also know that it sort of potentiates high frequency oscillations. So if you look at this plot, for example, if you have an increase in the low frequency oscillation, then you get this increase in the high frequency buzz on top of it. And then as that decreases, the high frequency oscillations decrease as well. And this is one way that people really think that you can have communication between distant areas, right? So with high frequency oscillations, the problem is that you lose phase information because they're oscillating too quickly. So how do you actually connect different brain areas that are a long ways away, where you still have to wait for the information to transfer from one location to another? And this phase amplitude coupling seems to be one possible mechanism for actually managing these long distance communication within the brain. So this is just an example of phase amplitude coupling in the human brain, so measured non-invasively with MEG. So you can see phase amplitude coupling shows as this pattern where you can see in different regions, and each of these different plots is corresponding to a different color and a different region in the brain. So even though the phase amplitude coupling, the frequency, the low frequency versus high frequency coupling is different in different brain regions, you can see broad phase amplitude coupling across the brain. So, so we know this is just an intermediate summary. So we know that alpha oscillations are strongest in the visual and somatosensory system when they're at rest. And we also think that this is also related to directing attention. So what we first measured was just this very simple situation where you close your eyes and alpha oscillations increase. There's more information now, or more evidence that alpha oscillations are also related to directing attention. So how do you decide whether to listen to the sound in the background or pay attention to the talk that is being given now? How do you decide that, and how do you suppress the information that you don't want to attend to at a given time. So this was a very complicated paradigm where they presented both visual and auditory stimuli. But the important thing is what they found is that you have either increases in alpha oscillations in the auditory cortex if you're told to attend to the visual, or you have an increase in alpha oscillations in the visual cortex if you're told to attend to the auditory information. So again, alpha oscillations seem to be a mechanism by which you can control your attention. And this is also correlated to behavior. So this was showing the kind of broad correlation between alpha amplitude and the reaction time in this task. And so you can see that the strength of the alpha oscillations, how well you are able to increase the alpha oscillations in the region that you were supposed to be suppressing, and how quickly you can respond to stimuli. There's another study that was really looking at the relation between occipital alpha power and attention. And this is our study, our one study that was looking at infants. So again, looking at neural oscillations, we're trying to understand basic brain development. And there were two different groups in this study. We had individuals who were exposed to alcohol prenatally. So there's a bunch of preclinical studies, studies in animals that have demonstrated that that the brain structure is altered in individuals who are exposed to alcohol prenatally versus those who are not. So the question is can we identify differences in infants who've been exposed to alcohol prenatally relative to typically developing controls? So we had 119 or 111 who were initially eligible to participate in this study, and we had 69 subjects who were analyzed for this study. What we found was in left hemisphere, we had kids with prenatal alcohol exposure, PAE, who had increased spectral power, and this is across the different frequency bands. I've been focusing on alpha and gamma, but these different frequency bands are all related to different functions. And in this case, the kids with PAE just have increased power relative to controls across the different frequency bands. And this is consistent with previous EEG literature that was from the 1980s. It was a long time ago, but similar things were found in the EEG literature. What we also found was that that amplitude, and this is just an example from one of those frequency bands, was related to the amount of alcohol that the infant was exposed to. So there was a dose effect. So we know from these studies that maternal factors influence in brain development, and especially prenatal alcohol exposure changes brain dynamics in infants. We've also done studies in kids with fetal alcohol spectrum disorder. So this is older kids. This was looking at kids in the adolescent time frame. And in this case, we focused on gamma power because, again, this was looking at multisensory integration. And we found differences between our kids with FASD and the healthy controls. And interestingly, this was also associated with latency of the M100 response, which is that initial activation of visual cortex. So we do think that the gamma oscillations are altered in kids with FASD, and there's broad literature kind of describing why that might be the case that we can't go into today. But it's also related to that very early visual response. We've done multisensory integration study in kids with FASD. In this case, we were integrating information from both the auditory cortex and the somatosensory cortex, so sound and touch. And this was another somewhat complicated paradigm, but essentially we presented auditory and tactile stimuli, and then simultaneous auditory tactile stimuli in both congruent and incongruent conditions. And again, we found differences in gamma oscillations in these multisensory conditions. So these neural oscillations also predict behavior. So the gamma oscillations were related to behavior on this extra dimensional shift task. So the number of errors that happened on that task were significant related to the power in the gamma band as well as in the beta band. And I haven't focused on beta oscillations yet, but this is another frequency band that is primarily related to movement, so motor cortex. You see beta oscillations that are active when the motor system is resting, but these are modulated by whether the motor system is activated or not. And this was a simple task where you number the buttons, and then the kids had to follow the pattern and implement that. And it's interesting because we looked at youth, young adult, and older adults in this particular study. And generally percent correct, everyone does fairly well, although the youth are not quite as good as the adults. Their reaction time is slower than both of the adult groups. And how long it takes them to press the button is the same between the youth and the older adults, but fastest in the young adult group. But you see this beta oscillation in motor regions. And this was also related to differences in spontaneous beta. So I was just referring to the behavioral information. What is interesting is that the older adults actually stick out more than the youth, but the youth are still significantly different than the young adults. So this is just demonstrating how dynamic our brain is across development, across the age span. And there are a number of different things that we could talk about regarding this, but we're almost done. So I just wanted to finish this out. So this is again just showing associations between those beta oscillations and behavior across those populations. And I wanted to stop and give people opportunity to ask questions, and thank you for your attention.
Well, thank you for a very nice talk. So anyone with a question should just speak up. Harjeet, for example, has a finger raised, but I don't hear him. Your audio is off, Harjeet.
Can you hear me now?
Yes, loud and clear.
Okay. I suppose all mammals have similar brains. Is that correct?
If you look at the microstructure, then it looks very similar. Of course, the size is different. Yep.
Now, do the animals like monkeys and others have problems like dementia and Alzheimer's?
You can cause that to happen. Yes, you can cause that to happen. In some cases, so actually dogs are the best model for dementia. they seem to develop some type of dementia. And you can also develop models of dementia in various animal models.
And does that help finding out what the cause of these things are?
It's a critical piece so I would say that, most of the drug development that has occurred is because you can instigate similar patterns in the brain in animals and then see what, so we have in Alzheimer's, for example, we have neurofibrillary tangles, and that you can look and see what type of pharmaceuticals can actually go after that pathophysiology.
Different species, dogs, monkeys, humans, eat different diets. Is there any connection between diet and severity of Alzheimer or dementia, and at what stage those things appear?
Yeah, there's quite a bit of evidence that that diet does play a role in dementia, because the primary risk factor for dementia in humans is cardiovascular risk factors. So if you have diabetes, or if you have high blood pressure, and these things are also related to diet. So eating healthy and exercising is still highly recommended for brain health.
I see. And does it have anything to do with the age factor?
That's complicated because, it appears that Alzheimer's starts in the brain probably 20 years before you actually see the behavioral manifestations. And because of that long lag between when it actually starts and when you can actually diagnose someone with having Alzheimer's, doing research in those pre-diagnosed individuals is complicated. So really the research is based on detailed history of, what people, just lifestyle factors that people are able to report once they're diagnosed. But, we also have perhaps not the best memory of, really detailing that carefully, looking back 20 years.
So the age doesn't.
Well, so age still has an influence as well. So if you look at risk of developing Alzheimer's with each decade after 60, then your risk increases regardless of any other factors. So there is still an aging component to it for sure. If you're above 80, you're close to 50-50 in terms of whether you're going to develop some type of a dementia.
Thank you.
Mm-hmm.
So let's see, maybe somebody else has a question.
I have a question. Thanks for the talk, Julia. So the chart you showed when you're explaining action potentials, the vertical scale was like tens of millivolts. Something I've always wondered whenever I see these biology related talks, is that the actual voltage you would measure if you had a voltmeter and could put it across the neurons, or is that sort of a signal they measured in the lab and somebody was a little bit lazy and backing out what the actual signal should be?
No, this is actual electrical measurements. So they do have the ability to do that with invasive techniques, and it's not my specialty, but it is. You can measure it directly.
Okay, thank you.
Mm-hmm.
By the way, let me just mention that that 50 millivolts is across 5 nanometers. And so the electric field is of the order of 10 megavolts per meter. In other words, this is a serious electric field, in fact, stronger than we normally have in the laboratory. Someone else would perhaps like to ask a question? I see Stefan Posse is here. By the way, I have perhaps a silly question. What does the damage to the body? Is it the ethanol or is it the antidote to ethanol? That is to say, when ethanol enters the body produces a chemical that neutralizes the ethanol. And in fact, the damage that's caused by drinking methanol is that the antidote to methanol is formaldehyde, which is much worse than the methanol. What hurts the human body, either the heart, the brain, or the fetus? Is it the ethanol or is it the, I forget the name of the analog of formaldehyde? It's anyway, it's acetaldehyde, isn't it?
It's not easy to answer that, right? Because again, these are, complicated systems. And even if you're working with the with an animal model, you can't go in there and measure directly all the time. But we know that ethanol is, it could be both, right? Is the answer to that. One thing that is a little bit interesting is that it appears that it's actually more damaging to have changes in ethanol content than it is to just have a status quo of ethanol. For example, binge drinking is more damaging than just having continuous exposure to alcohol. And so it seems to be partly the onset and of processing the alcohol and then the withdrawal of it and then the onset of it again that is actually more damaging to the neurons than just the presence of it.
But of course, in binge drinking, you have a very high concentration of alcohol, right? I mean, the 2 drinks a day guy is different from the binger.
Well, but you can have, people who have continuous alcohol that are much more than 2 drinks a day. So they're, comparing to, like to like, right? So individuals who are drinking at high doses.
Also, when you talked about closed eyes, I started closing my eyes, and what I've noticed always when I close my eyes is that I'm starting to go in the direction of sleep.
Mm-hmm.
So isn't that, I mean, it's more general than just don't pay attention to visual information. It's
well, so the alpha oscillations are only present when you have your eyes closed and you're not asleep. So the neural oscillations that happen during sleep are very different. And we know about different sleep stages, and those sleep stages are defined based on the neural oscillations that are present during the different sleep stages. So we actually have sleep quite well characterized as well, and that's primarily based on EEG studies. But MEG also sees the same type of activity. But yeah, it's this special little window when we have our eyes closed when we're not sleeping that you see those alpha oscillations.
I had a question about the alcohol study. So if I understand this right, the infants, they had this higher baseline of amplitude of these different, across the board. But then in the adolescents or the youth, it seemed like the amplitude was suppressed, or was it just because there was this higher baseline that you're
it's a good question. So there I can't really compare those. They're sort of comparing apples to oranges there, because the adolescent study was looking at the stimulus evoked neural oscillations rather than kind of the resting. So with the infants, we were looking at just resting brain activity, like you said, sort of the baseline activity. And with the adolescents, we have a separate study that actually looked at that resting oscillations in kids with FASD that is also different, but not different in the same way as the infants. So there are developmental changes in the pattern as well that we have identified. So
sounds quite complicated. Yeah.
it is. Yep.
Yeah.
That's the problem with the brain changing at the same time that you have other factors. So it's interesting because you can learn a lot about the brain with all these different variables.
Yeah. So is the goal to understand it well enough that you could potentially then treat it, or how does that work?
So I would say with non-invasive neuroimaging, there are sort of two primary goals. One is perhaps to facilitate identification, because currently diagnosis of different developmental disorders can be challenging.
Yeah.
And so maybe you can help with identifying individuals who have altered brain function because of some type of developmental disorder. And then the other possible goal is to inform treatment, right? So you can look at how the brain is now, and if you apply some treatment, does the brain look more normalized? Does it look more like a normal child? Or is it different? Is function better with that change? And, so we don't know if normalizing behavior will give them better function or not, but that's the other thing that you can think about in terms of how long-term non-invasive neuroimaging can contribute.
Yeah, it's fascinating. Yeah, thank you.
Yeah.
By the way, just to bring up alcohol and acetaldehyde again, my oldest son, who's a cardiologist, told me that when they do autopsies of alcoholics, they find that the cardiovascular system is like that of a baby, presumably because ethanol is a marvelous solvent and it just dissolves all the crud in the vascular system.
Oh.
and so I'm just wondering if there'd be some way to neutralize the ethanol and the acetaldehyde. This would be marvelous for people and be a fantastic commercial.
Yeah, I mean, the challenge with that, of course, is that if you look at the brains of individuals with alcohol use disorder, they're, they look much older than their age-normed counterparts.
Yeah, no, I'm not saying drinking is good for us. What I'm saying is that that it could be the acetaldehyde that does most of the damage, and it would be nice to have to have something that drinkers could take that would stop the ethanol, that would deactivate the ethanol and would also deactivate the acetaldehyde to prevent damage to the body.
Yeah, I agree.
Hangovers are caused, for example, by acetaldehyde, and if you could perform the function of acetaldehyde without the side effects of acetaldehyde, that would be quite remarkable. And it'd be a fantastic commercial product, but it would also do a lot of good, I would think.
Yeah, that would be amazing if you could improve people's cardiovascular health,
especially for beer, wine, and industry. Those industries would take off. Well, enough of my silly comments. Let's once again thank Professor Stephen for a very illuminating talk, and it's the kind of talk that I think we need more of throughout the American educational system, from high school to PhD and beyond. So thanks once again, and have a nice week. And I guess I don't know if I'm the one to stop this. Yeah, or, well, I can just leave. So we can all leave, and presumably Leanne will stop recording it. Anyway,
you.
thanks once again, Julia.
Thank you.
Nice talk, and thanks
Yep.
for putting up with my silly questions.
Take care.
Bye.